So in this lecture, we will be taking a look at the two senses that use chemoreception in order to send information up to the central nervous system. And those are the senses of olfaction, smell, and gustation, that's taste. So as a reminder, the special senses, and we're now looking at, the rest of this chapter is devoted to the special senses. The receptors are part of very specialized sensory organs that are all associated with the head. And they include the senses of olfaction and gustation, as well as hearing and equilibrium, that'll be in the next lecture, and then vision, which is in the last lecture. So there are two olfactory organs, and they're found in the epithelium that lines the roof of the nasal cavity. And these organs are composed of three types of tissues. There's the olfactory epithelium. These are supporting cells. And it's a columnar epithelium that they surround the olfactory receptor cells, and they have a yellowish brown color because of the pigment lipofuscin, which is abundant in the cytoplasm. Lamina propria is the areolar tissue associated with the olfactory epithelium, and it's got a rich blood supply as well as nervous supply, so we get the nerves that are leading away from the olfactory receptor cells are found here. And there is found secretory olfactory glands in the lamina propria, and they produce mucus. It's a fairly thick mucus with a yellowish color. And then last of all are the olfactory receptor cells themselves, and these are modified bipolar neurons that are very sensitive to dissolved chemicals. So we have a cell body. If you remember bipolar neurons, they have an afferent axon that brings information toward the cell body, and then an efferent axon that carries information back to, I got the right, afferent. Afferent brings it to the cell body, efferent sends it now off to the toward the central nervous system. So olfactory receptor cells are the only neurons in the body with their receptive surface in direct contact to the external environment. And so sitting way up here in the nasal cavity, they, as we breathe in, then, or as we eat foods, if the odorant molecules can aerosolize from the food, then they will make it up into the nasal passages as well. And when those odorant molecules get around that olfactory membrane, the chemicals then dissolve in the mucus that's produced by those Bowman's glands, by the olfactory glands that produce that thick mucus. And as the chemicals dissolve, then these non-motile cilia that are associated with the dendritic knobs of the receptor cells, the cilia have in them receptor proteins that will bind to specific odorant molecules. And so with sufficient binding, then we can get stimulation along the along the axons. We can get an action potential forming and transmitting information back up the olfactory nerve, that cranial nerve. The olfactory bulb is, notice that olfaction here is very closely, that olfactory nerve comes, and it makes it, and it is right here at the base of the cranium. We find this structure called the olfactory bulb, and then from that the nerves branch and come down to innervate each of the olfactory receptor cells there. So these are very sensitive cells. It takes very few odorant molecules to bind to be able to trigger an action potential. In other mammals, they are super sensitive. So with humans, we're fairly sensitive, but we are nowhere near as sensitive, say, as dogs or cats or wolves. So animals that really rely on their sense of smell. In those animals, like in our sheep, the olfactory bulbs can be very, very large. For humans, the olfactory bulb is a fairly small tissue. So the olfactory cells, because they're exposed, are relatively easily damaged, and so they have a relatively short lifespan. So they're replaced every 30 to 60 days or so. They're way up high in the nasal passages, and so they're out of the usual pathway of airflow. But if we do a sniff, one of those kind of drawing air, very heavily up through the nasal cavity, then that will bring air along with the odorant molecules across the olfactory surface. But anytime we have some sort of sickness that results in lots of mucus production, like a cold, then we can wind up really covering over. So as the mucus cells in the lamina propria produce lots and lots of mucus, if those if the cilia, the sensory cilia on the olfactory receptor cells, become really embedded in a thick layer of mucus, then it can reduce our sensitivity to odors and also really reduce the taste that we get from foods as well. As we get older, the number of olfactory cells will decrease, and those that are still there remaining become much less sensitive. And so, I'm suggesting that might be why Grandma really lays the perfume on so thickly, because that's just, she can't smell quite as well as a younger person is able to. So the pathways, the nerve pathways, the axons pass through tiny little openings in the ethmoid bone and make their way up into the olfactory bulb. And at the olfactory bulb, they join, forming cranial nerve 1. That's the olfactory nerve. Remember old OB, olfactory nerve. And these axons synapse with interneurons there in the olfactory bulb. And the interneuron axons then form this olfactory tract that terminates in the primary olfactory cortex, associated with or found there along the primary sensory cortex. Other interneurons will synapse, and then those interneurons are part of the limbic system, and so smells are very strong. We have strong associations of smells with memories and with emotions. Both of these are characteristic functions of the limbic system. Okay, so that is the one more bit. Okay, so olfactory stimulation. So this is the only sensory stimulation, by the way, that doesn't have to go first through the thalamus, but goes directly to the cerebral cortex without having passed through the thalamus. And with some of these receptors, as few as 4 odorant molecules may cause the olfactory receptor to reach threshold. So, so these are super sensitive to smells. And but olfactory stimulation also adapts very quickly. So if we are continuously subjected to the same smell, we can see reductions of 50% after just 1 second of stimulation. So we kind of move away from noticing smells fairly rapidly. We are capable of distinguishing among 2,000 to 4,000 different chemical stimuli. So that's pretty impressive. Lots and lots of different sensory receptor types, odorant molecule receptor types that allow us to do that kind of that kind of distinguishment among the stimuli. And what we get is that the central nervous system then responds to the pattern of the olfactory receptor types that have been stimulated to recognize specific smells that are characteristic of specific products or materials. So it's the pattern that, how many of one kind of receptor plus how many of other kind of receptors, what's the ratio of the different chemicals that we're able to sense, what is that ratio like among those different chemicals, and that tells us then, or allows us to distinguish amongst the different smells that may be presented to the body. Okay, so now that does it for olfactory stimulation, and let's move on then to gustation, to taste. So the tongue, of course, is the primary sensory organ associated with taste, and on the tongue we find taste buds. Now, I thought all my life, until I started teaching this class, that the bumps on my tongue were the taste buds themselves, but that is not the case. The bumps on the tongue are the papillae. So here's a papillus, and along the sides here of the papillae, that is where we find the taste buds. So a taste bud, a single taste bud, which is, this kind of material, all this here is a single taste bud, is composed of two primary cell types. There are the taste cells, that's what the yellow ones are, and then the basal cells, the supporting cells. And we find these taste buds along the sides of the papillae of the tongue, but also on the soft palate, way up at the roof of the mouth. There are some scattered along the inner walls of the cheek, and there are even some in the pharynx and back at the epiglottis. The epiglottis, recall, is the little flap of tissue that covers over the trachea, so that when we swallow, food tends to slide back toward the esophagus rather than accidentally getting inhaled into the trachea. And so the adult human has approximately 3,000, give or take, taste buds scattered across different areas of the mouth. But by far the greatest accumulation of them is associated with the tongue and the papillae of the tongue. Okay, so let's talk then a little bit about the taste bud cells. So first are the gustatory, or the taste cells. The taste cells, they're kind of elongated, and they have at the end of them microvilli. So these are folds then of the membrane of the taste cell. And these things are referred to, these microvilli are referred to as the taste hairs. And they project through a little opening here among the cells called the taste pore. So they're right there at the mouth of the taste pore. And they are then bathed in the fluids of the mouth, the saliva of the mouth. And embedded in these hairs, in the taste hairs, are receptor proteins that will receive specific molecules. And we find that there are coiled up around the taste cells are sensory neuron dendrites, and they are stimulated, these dendrites are stimulated as receptors bind to the chemicals that are dissolved in the, or as the receptors bind the chemicals that are dissolved in the saliva. So receptor proteins here, a chemical comes along, it binds to the receptor, that then causes some changes in the membrane, and coiled around here are the dendrites. The dendrites notice those changes, and if they receive sufficient, or that produces a local potential change. That if that neuron, the taste neuron, receives enough local potential change, then it will stimulate an action potential. The taste cells, like the olfactory cells, are relatively short, are very short-lived. The olfactory cells are relatively short-lived, but taste cells are very short-lived because of all of the friction and mechanical forces that we find in the mouth. And so they are replaced about once a week. We get a whole new set of taste buds. So associated with the taste cells are basal cells, here the supporting cells, that produce new taste cells. They produce those new gustatory cells. And so they divide by mitosis to replace the ones that are damaged as they are damaged. So taste sensitivity is quite variable among individuals. And it is certainly greatest, or sensitivity to taste is greatest in young people, and it declines as we age. And I suspect that this has a lot to do with why children tend to be particularly opposed to kind of bitter flavors and bitter-flavored vegetables especially. So, I never, ever liked Brussels sprouts as a child. They've got that real cabbagey kind of bitter flavor. But now that I'm an adult, or have been an adult, I love those dang things. They're so tasty. And so it is probably because of the reduced sensitivity that I experience in, with my taste sensations. So taste buds are all very, physically similar, but they are specialized for one of 5 basic taste sensations. They're sweet. That's what we get from carbohydrates, especially the mono and disaccharides. Alcohols have a sweet flavor. And there are some amino acids that also have a sweet flavor, as well as salts of lead, interestingly enough. So we have sweet taste sensations. Sour is provided by when we have sour taste buds encountering hydrogen ions in solution. So acidic stuff tastes sour. Think lemon or lime juice, for instance. Salty is that sensation is produced by metal ions, and I know it doesn't make sense to think about sodium as a metal, but in the chemical world, though, sodium is considered a metal. So sodium has the saltiest taste, but other ions can produce salty sensation as well. So potassium does this, and and when you look at the sodium-free salts, potassium chloride, KCl, is the primary one. But people who are on low sodium diets will tell you that you don't get as good flavor from potassium chloride salt. Bitter is produced by alkaloids. That's a special kind of chemical that really gets the bitter taste buds activated. So nicotine, caffeine, morphine, quinine, strychnine, these all have really bitter flavors. And even some non-alkaloid chemicals, like if you've ever eaten acetylsalicylic acid tablets, so that's aspirin. Even though it's an acid, it has a terribly bitter flavor if you ever just crunch down on an aspirin tablet. Then those are the 4 that we're probably most familiar with, but there's a 5th, only sort of discovered probably about 30 years ago or so. It is referred to as umami, and that is the umami is apparently a Japanese word that means delicious, and so it is a taste sensation that is triggered by amino acids and aspartate. So that's a chemical that is closely related to amino acids, and we find these umami flavor molecules in meats, in aged cheeses. So if you've got a really fine aged cheese, like Parmesan cheese or Parmigiano Reggiano, or those that have been sitting around in caves for a long time, they will have lots of umami flavoring in them. And it's the reason why a lot of Oriental restaurants really go overboard on the MSG, the monosodium glutamate, is because that provides a lot of umami flavor. And that's what, if you use the flavor enhancer Accent, I think is the brand name of it, it's just monosodium glutamate, and it gives this umami flavor to foods. Okay, so those are the 5 basic taste sensations. But they are, and it's not just those 5 flavors, though, that give us the taste. Olfaction has a lot to do with increasing our sensitivity to different kinds of flavors. And so, if we get that cold that's really knocking out our ability to smell, then it really mutes our taste, our flavor perception as well. Gustation is important because not only is it pleasurable, all the yummy foods that we eat, but it's also protective. So the reason that we experience such a strong reaction to bitter flavors, those alkaloids that we were talking about, caffeine and strychnine and morphine, these are all very harmful chemicals that can really mess up homeostasis. And so bitter especially is a good clue that something is really, really amiss in the food that we're eating. We are way more sensitive to sour than to sweet or salty. And again, there's that protectiveness where acidic materials, foods, could be damaging to the tissues of the body, of the mouth and the digestive system. And then we are even more sensitive to bitter than we are to sour, because it's 100 times more, more intense, or we need 100 times less stimulus to get an action potential than even sour requires. And again, that's because of the toxic nature of many of those alkaloids, the biological toxins. Okay, so last bit is to talk about the pathways, the taste nerve pathways. Taste buds are carried the sensations that are being produced at the taste buds are carried on one of 3 nerves. The facial nerve carries about 2/3 of the tongue's taste sensations. The glossopharyngeal nerve picks up the posterior third of the tongue, the taste sensations from there. And then the vagus nerve carries input from the walls of the mouth, the cheeks, and from the pharynx. And these nerve fibers then synapse with nuclei in the medulla oblongata. So we carry all of that taste sensation is carried back to the medulla. There's synapsing with the nuclei there. And then from there, the impulses are transmitted onto the thalamus, and then from the thalamus up to the primary sensory cortex of the brain. Okay, so that's everything that I have to tell you about taste and smell. So be sure to study well and contact me if you have any questions.